Hey guys, welcome to Unknowns and welcome to our exploration of fascinating history and functions of operons in molecular biology. Today we will be looking into lac and tripoperons. Okay, let's unravel the journey of this genetic control system that shape bacterial metabolism. The term operon has been coined in 1960 from this paper. Okay, by Jacob. He is the first one to coin the term operon. However, subsequent discoveries revealed that complexity of gene regulation unique to each operon. Okay, initially the Jacob uh, he meant that group of genes that is coordinated to perform a function can be the clusters can be called as an operon. So here comes a lac operon, genetic marvel utilized by E. coli for lactose metabolism. We know lac operon through studying E. coli's metabolism. It embodies the interplay of regulatory lac repressor, catabolite activator protein responding to glucose and lactose levels. Within lac operon, three coding regions, lac Z, lac Y, and lac A, produces enzyme responsible for lactose degradation and uptake. When lactose is present, it acts as an inducer, rendering the repressor inactive. This triggers RNA polymerase binding to the promoter, initiating the transcription of the structural genes. Now, see, it's a degradative pathway, but tripoperon is about synthesis of tryptophan. So, in tryptophan, which encodes the enzyme, the tripoperon encodes the enzyme tryptophan biosynthesis or tryptophanase or tryptophan synthesizing enzymes in E. coli or in general in bacteria. It's a the tryptophan regulation involves a repressor responding to the tryptophan levels. Okay. So the tryptophan comprises of structural genes, a promoter, a operator sequence, and termination region. The tryptophan is finely tuned to control tryptophan production based on the cellular needs. While the lac operon deals with the sugar catabolism, the tryptophan orchestrates the amino acid anabolism. Lac activation hinges on the lactose presence, whereas strip deactivation relies on the tryptophan abundance. And here is the general difference between the lac operon and tryptophan. These operon showcase the intricate dance of genetic regulation, influencing the cellular metabolism in response to the environmental cues. Okay, in an see, usually the operon stands as a captivating regulator, orchestrating the genetic activity with the precision and complexity. So these are the known aspects of trip and lac and trip. So in corresponding with unknowns, what we are doing, what is happening with this operon recently, meaning in 2018, this paper has come, which aims to explore the impact of transcription rates and the mRNA production in both active and inactive states of lac operon. Considering the complexities of inducer and repressor complexes, this interaction with the uh, not only the repressor complexes and their interactions with the DNA operators. This investigation also seeks to bridge the gap in comprehending how single positive feedback mechanism as observed in lac operon may extend to more intricate genetic networks involving multiple interconnected components with varying feedback controls. This is a very interesting article. If you see what the study focus on is how the binding affinity of specific inducer repressor complexes influences the feasible ranges of transcription rate necessary to achieve a steady state gene expression in both active and inactive states of operon. Yeah, ultimately, this research article aims to provide insights that could be potentially contribute to synthetic biological application and the understanding of genetic synthesis limitation. This is the beauty of unknowns. Even though we are studying Lycopran all these days, still many more work has to be done. When it comes to tripopran, especially with this pathogen chlamydia, this article is intended to explore evolutionary dynamics and functional alterations within the tryptophan operon of chlamydia trachomatis, specifically focusing on ocular and urogenital variants. While urogenital variants are known for synthesizing tryptophan from indole using intact tryptophan synthase, ocular strains are characterized as an auxotrope due to the frameshift mutation in the TRPA gene, leading to the loss of TS function and reliance on the host tryptophan pools for survival. This research really investigates the selective pressure influencing the maintenance and evolution of tryptophan operon among different series strains. See, this research gap focus on elucidating the dynamic evolutionary mechanism, functional alterations, and adaptive advantages of mutation within the tryptophan operon of series strains, shedding light on the interplay between host pathogen interaction and bacterial survival strategies. This is another cool paper to read upon. And overall, thank you for joining us on this journey through intriguing world of operons. Stay curious and stay inspired.